Good afternoon, everyone. I'm Babak Parvez. It's truly a pleasure to spend the next 45 minutes with you. Unfortunately, we don't have one of our panelists. I was really looking forward to his comments and presentation because his company is at the cutting edge of the field that we're going to discuss at this panel, but they had a COVID exposure, so they're not going to be here with us. So this panel is about synthetic biology, and I'm an optimist about technology, and I think this is one of the key areas that might define the next few decades. So if I look in the past and look at what really has changed our lives in the past few decades, it's been heavily driven by applied physics. So a lot of things that we've talked about, whether this is a transistor or a communication link that uses photons. Applied physics was or has been the king in terms of moving the society forward. A lot of inventions have come out of applied physics. It may or may not be the case in the few decades that are ahead of us. And one of the bets, at least I'm placing, is that there will be other areas that will be at least equally important. One of them is synthetic biology, and the other one would be perhaps applied mathematics as manifested in artificial intelligence. But synthetic biology has the promise of being a defining field in the coming decades. It's yet to materialize all of its potential. I think it has a lot of potential, so we see a lot of interesting things there. Many of those things have not translated to real impact, immediate impact, but we'll discuss some of those topics at this panel. So I have joining me two very interesting individuals at the forefront of what's going on in synthetic biology. The first one is Georg Seelig, so I'll invite him to come up to the stage. Georg is a friend, and he's a professor of electrical and computer engineering and computer science at the University of Washington. And what's really interesting about Georg is that he's showing this transition going from applied physics or physics to biology, because his PhD is actually training as in physics, and I believe he did solid state physics, yeah, at a very prominent group. And then he just completely decamped and went to Caltech and retrained as a biologist. And he's done some very interesting work in this field. And we're talking about entrepreneurship in this conference. So he spun out multiple companies as a result of his work. So I'm very much looking forward to his comments. And the next panelist is Matt Schultz. He's familiar to many of you. He was here last time. Another good friend, and someone who's also at the forefront of trying to engineer biological systems to do what we want them to do. They may resist actually doing that engineering, but I'm sure Matt is gonna talk about that. So the way we would run the panel, it would be a little bit shorter. Georg will come to the podium and present for a few minutes, followed by Matt, and then we'll start the conversation. So Georg.
Well, welcome. I'm glad to be here and tell you a little bit about the type of work my research group is doing. So the very big picture, what I'm interested in is kind of learning to read and write the language of the genome. And so what does that mean, right? So we're all familiar with the fact that the genome is sort of a code. It's 3 billion letters that sort of specifies our hereditary information. And over the last, it's a 4-letter code as sort of shown in the image. And over the last, few decades, we've really learned a lot about the syntax of this language. We've learned about the words of the language, but we still can't quite put it all together, right? And you can ask, why do we want to put it together? And at least from my research, there's sort of two motivations that really drive my interest in this language. Like the first one is the observation that between any two of us, right, most of our genomes are similar, but there's probably about 5 million out of the 3 billion letters in this code that will be distinct. And if you look at the entire human population, like all 8 billion or so of us, probably every possible, single point mutation, like every substitution where you take, one of the typical letters and replace it with one of the other three, it's probably out there somewhere if it's not embryonically lethal, right? And the question then becomes, what do all these variants do? I mean, many of those might make us susceptible to some disease, others might make us more resilient. There's some interplay between them, and many of them probably don't do anything at all, right? And so this question of how can we actually learn and really interpret what these variants mean, I think is still a really challenging and open question. Now, the second part that's really more synthetic biology at its core is, of course, we don't only want to be able to really read and interpret this language, we want to be able to write in this language. And the reason we want to do this is because we want to be able to essentially design molecules, right? So we want to design, we want to design enzymes like the proteins, for example, antibodies. We want to design mRNAs and gene therapies. We might want to design entire cells, right? Cell therapy has been a really big topic in the last decade or so. And we might even want to engineer, cellular populations. We might want to engineer bacteria that can produce high-value chemicals for us that we currently make from fossil fuels, or that we could even, integrate in our gut microbiome and have produce drug molecules directly in response to something these bacteria sense in our environment. And so for all of these applications, what we need to learn is how to write genetic information. And I just want to give you one brief example sort of from the space of mRNA and gene therapy, right? So we're all at least familiar with these mRNA vaccines that have had such a big impact in the last few years. And if you think of an mRNA vaccine, right, it contains a part of it that encodes for the protein, but it also contains these leader and trailer sequences. And these leader and trailer sequences are part of the mRNA, and they essentially determine how much protein is made, where the mRNA goes, how stable it is, and all of these things, right? And if we really want to make a good mRNA therapy, we actually have to engineer these sequences, and we have to learn the rules according to which, these sequences determine how much protein is made. And so this kind of motivates this need to really rewrite the language of the genome, right? So we want to have a sequence, in this case, that makes a lot of protein, as opposed to little protein. And I just want to give you like a 1-minute overview, specifically how my research group kind of goes about trying to learn this language. So to that end, we kind of use synthetic biology in the sense that we use, we might make a big library of synthetic genes, right? The human genome has about 30,000 genes. We can make, millions of genes in the lab very easily. In this particular case, if I want to study, let's say, the leader sequence of an RNA, I might just randomly vary the leader sequence in my synthetic genes, make a big library, test this library in an experimental assay. In this case, I would measure at very high throughput, like for each of these sequences, how much protein is being made. Of course, with such a big and sort of well-controlled data set, I can then use machine learning, right, to learn the relationship between the sequence and the function that I want, which in this case is make a lot of protein or learn the rules of how much protein is made. And then I can apply these models to, again, the goal of variant impact prediction. I can even interpret these models to kind of understand the underlying biology. And certainly I can use these models together with like generative design algorithms to make sequences that maybe are better than the ones we currently have. And so just to kind of, sum this up, I think the next step, at least for me, in this sort of process of learning this language of the genome is to really understand cell type specificity, right? If you look at us, we're a fairly complex organism. We start from a single cell, and now we have, billions of cells and thousands likely of distinct cell types. Each of these cell types contains the same exact genome, but somehow reads this information differently. And really understanding the origin or mechanism of this cell type specificity, I think, is fascinating because it would allow us, for example, to engineer gene therapies that are much more specific, right? So we can target a cancer cell and minimize side effects in surrounding healthy tissue. So that kind of application really requires us, again, to kind of understand this language in detail. And just to finish, I also want to mention that, as Babak pointed out, my lab, obviously, we're an academic lab. I'm a big fan of academic publishing. But we are also trying to commercialize some of the things we do, and recently did a startup in this space of sort of single cell analysis that's been getting some traction. So with that, I just kind of want to sum up that what we're doing is we're using synthetic biology tools to understand the genome, and then we're kind of also using these same tools to kind of engineer novel type of therapies. So thank you.
Thank you.
Thank you. So I'm going to talk a little bit about maybe the business end of this stuff, and that I'm interested in applying synthetic biology in medicine. And you might remember Eric Schmidt talking about yesterday the desire to use these technologies to do manufacturing, to take cheap feedstocks and produce useful valuable chemicals and compounds. And I think that that's where a lot of the field's been going, but I'm interested in using these kinds of tools and techniques inside of living human beings. And I think it's worth pondering for a second where we are in the context of this and how medicine's changed over the years, because for really thousands of years, medicine was plants and prayers, small molecule drugs and rituals. And it stayed that way pretty much since the dawn of recorded history until really the 1700s. We started taking like the first kinds of vaccines or what we might consider a biologic today. And then it was another century or more before we started making recombinant biologics, where we'd actually change the DNA of a species with the goal of getting something out of it. And this has kind of continued to progress into the last really couple of decades, where we started to get the first cell therapies, from the old school bone marrow transplants to things like modern gene-modified CAR T cells. And when I got into this field, there were no approved gene or cell therapies. And now there are literally thousands of them in trials, and they're making billions of dollars for the ones that are approved. And so the real question then, I guess, is where do we go next? And I think one of the most exciting things in the field is really this impending marriage between the worlds of computer science and information sciences and synthetic biology. And so this is actually what got me into the field in the first place. The North Star for me was effectively that the essence of life is information, that chemistry is a substrate. And you can tinker with the substrate, but it's a little bit like trying to debug Microsoft Word by changing the microchips in your computer. It's fraught with unintended consequences. And that's why every drug you buy has a list of side effects that's longer than the intended effects. And so I wanted to be able to interact with life in its more native language. But it was pointed out to me fairly early in the journey that cells don't have USB ports. They don't like you changing their information. And so I had initially set out to make a company to build an app store for the human body. And it actually also took through Caltech. Some of the first technology was out of David Baltimore's lab. And we began to build tools that could manipulate the immune system specifically. And actually after 13 years, that first one just now got permission to enter human trials. It got an open IND a few months ago. But I've become interested in using this technology for a field in particular, in aging. And actually, when I was first messing with the App Store, the goal was to effectively try to recreate the biochemical environment of youth as you aged. And which might sound a little absurd, but if you took your blood, once a year from birth until death, it's not the same, right? Lots of things change inside. Some are obvious, but you can never take enough drugs to put it back the way it was. And so going after aging is something that's not really practical with small molecules, but it also has tremendous value in that age is pretty much the master corollary for any malady you can think of. I mean, you have far worse odds of having cancer as a clean living 70-year-old than you do a chain smoking 20-year-old. And this isn't to say that your ill-spent youth won't haunt you later. It's just that age itself is the dominant risk factor. And I know it's controversial in some audiences, but I don't think we'll ever be able to solve it with pills for the reasons I was describing earlier, that you just can't alter these processes with small molecules, at least not to the extent we'd like. And so we really need better tools if we want to address longevity and health span. And this led us to really create a longevity medicine platform with a very broad mandate to go after all sorts of age-related maladies by addressing them at kind of a source code level, if you will. And the real problem with this is that the body has a vested interest in not letting you change its DNA. Like, it goes back to lacking the USB ports again. And certain technologies exist to do this, viruses being probably one of the more obvious ones, and things like lipid nanoparticles that were mentioned before on the vaccines for COVID. But they both have real limitations in that if you can't administer lots of these more than once. And so we wanted to build a tool that would combine the best attributes of the non-viral systems with the viral systems. And we built this cool proteolipid vehicle, and I don't have time to go over it, and I won't bore everyone with the technical details, but it has a couple cool attributes. And one is that it's kind of, like I said, it's a hybrid between a lipid nanoparticle and a virus, and it has a little fusion protein on it from a virus that used to infect the stomachs of alligators and birds. And it's the smallest of its kind by more than 100-fold, and it can be effectively invisible to the immune system. But it allows us to effectively just fuse with the cell and drop the payload inside. So it takes targeting really out of this realm of chemistry and out of the world of endocytosis, where it has to be taken up and confused with membranes non-specifically. And it allows you to do some pretty fun stuff. I'm going to keep track of time. This was a project we've been working on for a while to build muscle without exercise, basically. And there's a theme in some of my research. Centers around vanity and laziness. But these mice were given one treatment of this, and a year later, they're about 50% larger and twice as strong. And this, I think, has real value in that as you age, physical strength is everything. But it's a real big difference between something like, say, an anabolic steroid or a hormone. Like, this is effectively the biochemical pathway for skeletal muscle exercise. We wrote a program that says, tell your body you're working out when you're not. And that's something that you couldn't do before. And I think it's a really kind of illustrative of what we're doing in the field. And the other thing you can do that we've played around with a lot is you can kill things. And because you can target now with a computer program instead of poisonous chemicals and surface markers. And this is just an interesting example. The mouse there on the left was injected into its IV tail vein with genes that encode firefly protein, the thing that makes fireflies light up. And you can see the mouse is lit up there on the imager, and all of its organs are lit up. But on the right there, that mouse got the same dose, administered the same way, but you only see that little area in the red dashed line lit up. And inside there is a tumor that implanted in it. And so we've been able to get the expression of this gene restricted entirely to the tumor. So the idea that you could kill a cancerous cell based on what it's thinking, not what it looks like or what it's doing at the moment, but what genes are being read, what are on and what are off. And this all kind of moves towards building more and more complex circuits. So we started playing with a tool to kill fat, and that's took people that had tummy tucks and kind of punched the skin out, little cookie cutter style, and injected it into it, and you could kill about 20% of the fat cells with a treatment on it. And we think also goes back to my vanity, not wanting to do sit-ups, but these kind of precision level of targeting is something that you couldn't accomplish with technologies in the past. And I think it's really where the field is going now. And as this kind of loops back into the world of synthetic biology, it's building more and more complex circuits. So this is an example of making CAR T-cell therapies inside of the patient. Instead of having to take the cells out, you can drop the cost of goods from hundreds of thousands of dollars to the cost of a biologic, basically. But the rate limiting step is how much we understand about the control logic of life. So all these, although they sound pretty amazing, they're all use relatively simple genetic circuits. And so my hope, and what has me the most interested about this panel and synthetic biology in general, is basically bringing the information sciences to bear in developing ever more complex circuits that we can use tools like this to upload to people. Stop.
All right, thank you very much. All right, so this was a pretty interesting spectrum, starting from basic questions of how we understand this language, transitioning to how once we understand how we write it, and then once we have the ability to write in this new language, how do we use it actually to do something that people want to have. So, for the benefit of the audience, I want to ask you a question about the evolution of the field in the past few years. Maybe you look at the past 5-10 years. And you talked about some of these examples, but if you could share more with the audience, I think that'd be good. So what is it that we can do today in synthetic biology that 10 years ago we could not do? So if each one of you could please sort of take one or two items that today we can do it, 10 years ago we couldn't do it. If you could share that with us, that would be great. And then tell us, why is it that now we can do it, but 10 years ago we couldn't do it? So either Matt or Georg, whoever wants to start. Georg, you want to go first?
Sure. I mean, let me step back a little bit and just say something about what I think synthetic biology is, because that helps us kind of understand, where we are and where we want to go. And to me, synthetic biology, it's an engineering discipline, so we're trying to build things according to a specification. So it's not discovery science per se. And what we're trying to build at its heart is essentially gene circuits, right? Like as I think I mentioned, like to me, the most impressive set of, okay, maybe what's a gene circuit, right? So gene circuit is a collection of genes that talk to one another and process information. And maybe the most impressive set of gene circuits is really the circuits that drive development from a single cell to developed organisms, right? And it's many genes, they get switched on and off, they talk to one another and somehow control, not only information, but also spatial arrangement of matter and molecules. And synthetic biology, to me, started in 2000 when two papers came out that essentially, for the first time, kind of made these like synthetic gene circuits, and they're really kind of toy models. Like one of them was sort of, two genes that mutually switch each other off, so they kind of make a toggle switch. And the other one was essentially three genes that are arranged in a way that, first one switches off the second one, the third one, the third one, the first one. So it's kind of like a rock, paper, scissors kind of system that essentially oscillates in time, right? And these systems really captured people's imagination because they were just the first time we saw that, you can take these genes from different organisms, stick them together and make something functional, right? And I would say over the last 20 years, we've really learned to build systems that are much more accurate than those systems were. Because in the beginning, like this, these oscillators, for example, they didn't really oscillate at all in 40% of the cells, and probably in the ones where they did behave okay, like periods and amplitudes of these oscillations were all over the place. Nothing like what we're familiar with in electrical engineering, right? And so nowadays, I think we have a much better understanding of how we can like fine-tune these genetic components to actually make them work together. And I think the other big breakthrough that I just want to, it's a bit of a non sequitur, but I think that's also super important, is just the arrival of gene editing techniques like CRISPR-based gene editing, right? That really kind of transforms how we can manipulate information that's already there in the genome.
So it seems like our ability to manipulate has substantially increased, but is that because we just got lucky since someone discovered CRISPR? Or was there a more systematic way to get to this point that now we have much more capability compared to a few years back?
Well, CRISPR is interesting in that it didn't fundamentally allow you to do something that couldn't be done before. What it did was make it cheaper and easier and faster. And this, I think, kind of goes to the way I look at this question. Admittedly, more translational than academic, but the big advances have been ones of scale. Like the cost of reading and writing DNA has come down faster than Moore's law. And like right now, you can type a gene sequence into an iPhone and it'll show up in the mail in a FedEx envelope. Like you can sit there writing programs in the language of life. And that's amazing. Like you couldn't do that before. And CRISPR is one of these things that you can literally just order any CRISPRs you want. Like there's people selling kits to CRISPR frogs right now that you can do at home quite literally. I'm not advising it, although it is super interesting. Really the accessibility and cost of the tools, the efficiency of them is, I think, what's driven the most growth for my purposes.
Yeah, I agree with that. I mean, I think DNA synthesis and sequencing really kind of have transformed what we can do, and I think it's still going. It's going to get cheaper. There's more synthesis companies and more sequencing companies coming online, so that process is still ongoing. I mean, I also think there is sort of been just progress after having seen that. Look, hey, we can build some synthetic gene circuits, and they're kind of cool. I think there really has been an effort at understanding how we can more systematically engineer them and also characterize them and use computational, like CAD-style tools to put these circuits together. So I think it's not random chance. I mean, it's intentional.
So how close do you think we are to like a DNA compiler or an integrated development environment for this kind of code?
I mean, we have a few examples of that, right? I mean, Chris Voigt's lab, for example, built some sort of compiler, or at least like a Verilog-style programming language for DNA, for certain types of DNA. I mean, I think we're still quite far away from like sort of a general purpose compiler. I think in certain settings, maybe for the particular metabolic engineering, meaning that the process where we put genes together in a bacterium or so to produce molecules for us, we're maybe getting a little bit closer, at least sort of in the context of E. coli. But I think there's still a lot of exciting stuff to be done for like a general purpose approach.
So it seems like there's consensus that one of the major enablers of these advances in synthetic biology has been the reduction in cost in DNA sequencing, and also our ability to synthesize longer and longer strands of DNA. A lot of it is driven by automation and robotics, I would say. So when do we get the same thing for polypeptides and proteins? What does it take to get the same sort of Moore's law improvement for proteins? Because if we're just sticking with DNA, that's just a portion of the story. So we need to kind of graduate up.
Yeah, so I think it's a very good question. So basically, right, the progression is kind of like it's DNA. DNA makes RNA. RNA already tells you a lot about what's different, the different cell types and different organisms and whatnot. And then RNA, or many RNA, get translated to protein, and the proteins are really the thing that do most of the actions inside the cells. So right now we're very good at reading out RNA, even at the single cell level, but reading out proteins is still much harder. Now, personally, I think protein sequencing is very close. Like there's a whole bunch of cool technologies out there, like either based on sort of nanopores or single cell microscopy type of single molecule microscopy type approaches that, in my opinion, are less than 5 years out from really transforming this field of reading proteins.
We're going to hold you accountable to that prediction of 5 years for protein sequencing. So, Matt.
I've outsourced protein production to the patient, so I'm mostly on the writing end of this, not the reading end of it.
All right, so let's move to the applications now. As things are today, when you look at very broadly at synthetic biology, and we're going to start with medical applications, and then we'll talk about non-medical. But let's start with medical. As you look at synthetic biology today, what is it that you find most striking? What is it that we can do today with synthetic biology that you find most striking? And striking is your own definition of striking. It could be the most useful or unusual or controversial, but what is it that this audience should know about that you find particularly interesting in synthetic biology today, applied in medicine?
Well, I'd say that there's not much synthetic biology applied in medicine today, that this is largely something that's coming and close on the horizon. But so these genetic circuits that I was showing in the slides are far too primitive, in my mind, to be called synthetic biology. And the more complex circuits that are being built have not been deployed medically. And so I think that we're kind of on the threshold, and there's a lot of reasons maybe we're not there yet, and a lot of reasons we might get there soon. But we're on the precipice of another significant advance when this goes in. But I actually don't think it's made much headway into medicine yet.
And to some extent, I agree with that. I mean, I think the beginning of a counterexample of it is sort of the CAR T cell or the cell engineering world, where I think early CAR T cells or current CAR T cells are essentially just you modify one protein or one receptor inside these immune cells to kind of arm them better against cancer or some disease.
Actually, I apologize for interrupting you, but I think it's beneficial for the audience. Maybe you can take a step back and just in 2 minutes kind of explain how CAR T cell therapy works. So I think just go through the flow of what is it that people do.
I'll try. So basically, you take T cells, typically from a patient, and you engineer them to provide them with sort of a cell surface receptor, or modify their cell surface receptor, such that they can essentially talk to other cells, sense other cells. And that ability, allows them to then, when they, you take them out of the body, you engineer them, you put them back into the patient. When you put them back, because of their sensing capability, they can essentially hone in onto cancer cells. And because they're T cells, they can then recruit sort of the immune system to essentially remove those cells that have been recognized, right? And so the synthetic biology aspect in that part, and I don't know if you even want to call it that, but is really just the engineering of that receptor, right? Because that's something that's different than it's in the T cell that you take out of the patient to begin with.
That's an interesting point. The CAR, like third generation and fourth generation CARs are quite a bit more complex than the first ones were. And I mean, you think the first ones were very simple. It's like stick to that thing and pump it full of poison. Like they did not have a lot of bells and whistles, and they still worked, I suppose, remarkably well and launched the field. But the new ones are a lot more sophisticated and basically mediating survival of the cell and how it behaves beyond simply killing something. But yeah, I think it's an interesting, I guess, pedantic point about where do you cross the line from a simple like promoter gene setup to something that would be better described as synthetic biology. And it definitely, I think, there's kind of a gradient between them. It's not necessarily a really hard line, but as you start adding more and more logic at some point, you're like, oh, that looks synthetic biology. Like,
Right. I mean, just as a concrete example, right? I mean, there are now CAR T cells, at least, in academia, in academic research, where people are trying to sense multiple markers on so basically saying like, okay, if you just target one marker on a particular cell, maybe that marker is shared not just between the cancer cell, but also between other cells, and you don't want to kill those. What if you could look at multiple things, right? Sort of say this and this, but not the third thing, makes a cancer cell. And so this type of more complex logic that people are trying to engineer, I would call synthetic biology.
Yeah, I personally find it absolutely astonishing. Clearly, we couldn't do this 10, 20 years ago, to get my own immune cells out of my body, train them to do something that they don't do, put it back in my body and cure me from a disease. I think this is absolutely revolutionary, and we can do it today. Absolutely not doable 20 years ago. So this is a great example of progress. Debatable if you want to call it synthetic biology in a traditional way or not, but definitely, clearly, new capability to engineer biology to do something useful that we care about, and it works, and it saves lives literally. So we're creating these new capabilities that I can read this language. I can, to a degree now, write this language. I can create new therapies in medicine. But we kind of all know that what the flow is. So if I have a new technology or technique, it works sort of in the lab, then it has to go to animal testing, then it has to go to clinical trials. And as I get closer to real humans, it gets slower and gets more expensive. So it seems like the beginning of this trajectory has made a lot of progress, but it's all rate limited by what I can do with clinical trials, how much money I have, how much time I have. So how do we overcome this bottleneck? Because we have brilliant people like you here that may come up with amazing things in a laboratory, but if I don't have hundreds of millions of dollars and years to transition every single one of them through a clinical trial, then they don't see the light of the day. So how do I accelerate sort of the back end of this? So the whole thing from ideation until something is provided to patients can go faster and cheaper. So what are the good ideas there?
Go ahead.
Well, so part of it we've already seen, and that's really the progression in cell therapies. So from the first CARs or Immusoft's first B-cell treatment to the versions we have today, the tools used to grow these cells, to characterize them, to kind of control, manipulate them, have all just radically improved. So pretty much every tool we're using today didn't really exist a decade ago. I mean, it's all very modern tools and techniques, and that's speeding the process up quite a bit. I think the way to make it go even faster is to be able to make the change inside your body itself. And it's partly why I've begun focusing on this delivery technology. And I think it's really been the Achilles heel of genetic medicine is delivery. And so if you can take, let's go blue sky. I have a DNA printer, right? Which is a dream of mine. Like I want a DNA printer sitting there and I can type in any sequence I want and I can get the sequence out. And now with a microfluidic system, I can make a vector to put it into a person. That drops that cycle down to almost nothing. Like it really, we had a talk with the space medicine program a while ago about putting something like this on, the space station or on Mars and send a treatment wirelessly into orbit. And now the astronaut makes it in their own body. But that's I think the kinds of advancements, so I'll make it quicker. And of course that glosses over this little issue of the regulatory system that I'm sure has opinions about it. But.
Yeah, so let's talk about that. So how do we? So probably we're not that far from having a DNA printer. Effectively, when you can type the sequence on your iPhone or Android phone, I carry an Android phone. I used to work for Google. So if you can type the sequence on your phone, the next day with FedEx, your DNA arrives. It's not really that far from having a DNA printer, and we have the ability to put it in vectors. It's all there. But still doesn't solve the problem that new biological entity that's engineered by you has to go through a clinical trial before it can see the light of the day, which can take years and hundreds of millions of dollars. So we're still stuck. So at the beginning of the funnel, we can innovate and we can go very quick, but we have a massive rate-limiting sort of entity there between all your innovations and sort of real-life applications for patients. So how do we get this funnel to go faster? Like what changes do you want to see in the regulatory landscape?
More pandemics?
Definitely not, but what changes can make this go faster?
Right, so I really didn't want to answer this question because I don't really know the answer, but I mean, to me, it's fundamentally that is the big question, I think, in biomedical research. Like, why is it so expensive or why is it so slow for that last step, right? I mean, we have all these amazing things that work in the lab and in mice, and then they fail. And I mean, I think it's going to be, and in particular, I think this is a big challenge when you really want to personalize therapy, right? Because obviously, right now, if you do that something like T-cell therapy, usually you have to take cells from a particular patient, engineer them, and put them back in the same patient, because otherwise you get all sorts of immune responses that you don't want. I mean, to me, fundamentally, it comes down to sort of a lack of understanding still, that we just don't know enough about how our bodies interpret the information that we put in to really predict what's going to happen. I mean, I think it's not going to be easy, but if we really understood, how our immune system responds to particular perturbations, and we could actually accurately predict that, I think that last part of the cycle would be much easier, right? We wouldn't have these surprises anymore where a drug works beautifully until the point where you actually test it in people. And
all we have to do is understand life, right?
I think so.
Yeah, it's a piece of cake.
All right, thanks.
At least the immune system. Yeah.
Not anything that you want to say about potential changes to the regulatory landscape that would be sensible but still allow you to go faster?
Well, I do think that there's this fundamental trade-off between kind of safety and speed, and that if you, all these, the COVID vaccines were built very quickly, right? Like it didn't take long to design them. Like the one we made took us a couple of weeks. Then it was paperwork and getting into trials and making it and all this stuff. And so there's the intellectual capability to spit these out quickly, but it comes down to will and policy, basically. And it's why, you think of all the early gene therapies were really confined to lethal diseases, basically. Things that they said, okay, it's worth the risk to tamper with the DNA. And so I think as we become more comfortable with it, you think every year you get, the flu shot, they don't have to go through the same approval process every year, right? It's been kind of accepted that template is reasonable and they're changing the protein, but it's going to be okay. If we can get to this level of what amounts to like a master file with various genetic vectors, I think you could speed it up quite a bit. And I wouldn't say it's attainable, might be a strong word, but at least not insane.
All right, we're so rapidly running out of time. I want to ask you a couple more questions, and then maybe we can allow the audience to ask you. So let's talk about non-medical applications of synthetic biology. So real quick, anything you're excited about or you've seen in the field that's not medicine but synthetic biology that you find particularly interesting? Georg, you want to go first?
Sure. Yeah. I mean, I think in sort of metabolic engineering, it's a whole other level. I mean, people like Jay Kiesling, make like tens and tens of genetic modifications in bacteria to essentially program these bacteria to produce whatever it is that we need, right? And there's more and more examples. I mean, the first sort of SynBio example out there was it's now discontinued, but was essentially an anti-malarial drug that came out already in 2006. But nowadays we can produce almost anything from E. coli, and I think that's it's a technologically in a way a totally different level or it's possible to make much more complicated systems in E. coli than it is in human cells, maybe not surprisingly. And I think this idea of eventually replacing all these high value chemicals that come from petrochemicals right now with bioproduction, I think is really great.
So manufacturing molecules, as Eric Schmidt was talking about yesterday. Yeah. Matt, anything that you find interesting in synthetic biology, non-medical?
Yeah, I think it's mostly just the tools still. Like it's similar actually to the medical stuff, but that we're getting better at screening these faster. We're shrinking the error bars, able to understand a little bit more about what the consequences of our modifications are. And I mean, this has been the fundamental problem with this, is that we don't really understand the control logic of life. We don't quite know how it works. So we're confined to just breaking things and seeing what happens, or building a bigger library. And in some respects, we've gotten a lot of mileage out of just making tools that made it more efficient to build libraries and screen them. And so that's been, I think, propelling the field a lot.
Awesome. All right, last question, and feel free to sort of be controversial or just go out there. So we talked about what is it that we can do today that 20 years ago we couldn't do, and we've come a long way. So if you sort of mentally fast forward 20 years, what do you think that we can do 20 years from now that today we cannot do?
Yeah, I mean, I really think that we'll see a totally different level of programmability of organisms. I mean, again, right now we're building systems in human cells with 2 or 3 genes that we modify. Development, is driven by thousands of genes in a very complicated process. I mean, I really don't see any reason why we wouldn't learn to, write programs of that level of complexity with hundreds of genes driving, complex multi-stage processes, interfacing with the immune system. So I think a lot of what we're going to see in the next 20 years is like a totally different scale at which we can operate, and a much more finer-grained understanding that comes from this sort of combination of better measurement tools and better machine learning approaches of the code that sort of drives life.
I have a controversial one for you. I think we might be able to shift the balance of power in medicine back to the patient. And that, we're in a golden era of, for lack of a better word, biohacking right now. And I mean, if you think back to the world where before the U.S. became a really powerful material science group, you could get a chemistry set for a kid that had radioactive elements and instructions for building a bomb. And this launched a whole generation of material scientists who turned the U.S. into the world leader in this for a time. And now if you get a chemistry set, it's got baking soda and vinegar, and no one cares. It's way less interesting than an Xbox. But right now there's this ability to say order DNA online and getting cheaper and cheaper vector production is really going to allow people to actually design and build and test on themselves, most likely, treatments that would have never flown before. And I expect there will be consequences and backlash, and kids will lose their eyes like they did with the chemistry sets. But I think that is something that the world is not quite ready for, and it's already on top of us. So you can buy your kits to CRISPR creatures in your house right now. And so in 20 years, what's missing on that is a really easy tool to put it into you. But that's not hard, actually. That could easily exist then. It exists today, just not in the paws of the average biohacker.
Thank you. Steve, I don't know if you have time for any questions from the audience.
Yeah, we do. If there are questions, we can
so let's open the floor.
Yeah.
Hey,
Gretchen Huizinga. My interest is AI ethics from a religious perspective, and this raises all kinds of questions. Ray Kurzweil's The Singularity is Near, he makes a statement in the introduction, in the future we'll be able to live as long as we want. And then in parentheses he says, a subtle distinction from forever. And so my question to you is, how long is long enough? What's your ultimate goal? And what does the world look like if you're wildly successful and you've got all these old people who, don't want to hang up the spurs? And then so we've, back to Malthus.
Well, I suppose, the question of how long you want to live probably changes with time, because, maybe not, I don't want to die today. Tomorrow I probably won't want to die either. And so you can see how it would slip. But I think it's worth considering that if the tools we're building were to work, you wouldn't find the world full of 80 or 90-year-olds forever. You'd find it full of 30-year-olds forever. Like the idea that you could, if you can actually go after aging and make a person to their younger form, they're not going to be packing the retirement centers anymore. Like you're going to have a world full of young people. And I think that obviously if you just keep having lots and lots of people and keep living longer and longer, the population will change. But already people are having kids later and later. And so the counter argument is like, if I could say you'd be 30 forever, or until, you get hit by a car, when would you have your first kid? And it's going to, if you talk about especially if you apply that question to women, not men. And so it would radically alter the behavior of people as well. And so I don't think that you're running into a Black Mirror episode full of 90-year-olds and eating Soylent Green or something.
Please, go ahead, John.
Okay, John Wallstead of Discovery Institute. A quick comment and then a question. The comment is we may discover that the only thing that might be worse than a finite lifespan is an infinite lifespan. I don't know that we're prepared for what that means. But the question I have is, this is all very interesting, and what's going to happen when the Chinese put it in some lab in Wuhan and decide to work on it?
That was a softball.
Maybe I'll take that because honestly, we have no idea what they want to do. And we don't have any control over them.
Except that it's going to be bad.
We don't even know that because we just don't have control over that system. We don't make decisions for that system, just sort of independently running.
It is also worth noting that the U.S. occasionally kills biosafety level 4 scientists with its own creations as well. And so this is really, I think, more part and parcel. It's a risk of the technology. But engineering superviruses is kind of a dubious proposition to begin with. Making a mouse that gets 10 times as strong is probably not going to wipe out 1 million people or anything. It's just one unhappy technician.
Okay, Steve, let's take one final question.
So but what could wipe out 1 million people is a mandatory vaccination program where a genetic program is inserted into and required to be put into every individual, and then that code was somehow maliciously created, whether it's whoever the source might be. So that, from an ethical standpoint, the potential for good is tremendous, but obviously the potential for bad.
We're coming back to where we started: good versus evil, biomedical issues. I think we're going to.
Sorry.
Go ahead, Georg. Go ahead.
I think this, is a fair point. It's just not very specific to bioresearch, and it's not that'd be a very complicated way of killing off a lot of people. And there's many ways in which we can kill a lot of people right now. So it's not to say that we shouldn't think about the potential of bioresearch to have negative impacts, but it's similar to all technologies, right? Artificial intelligence has all sorts of potential, negative uses and positive uses that are not, it's not any different from bioresearch. And in some ways, it's this work is going to happen, and if it's going to happen out in the open, people can see what kind of research we do, we can publish it, people can double check it. I think that's actually probably the safest way of doing it. as opposed to, maybe trying to make it illegal and then it will only happen in some, lab hidden in some other country, but it will happen anyway. So I think that's, worth considering.
It's also super hard to make technology that works that well in general. Like everyone's always worried about, what if we can just deploy this everywhere? And it's devilishly hard to make these things function. Period. Although I shouldn't downplay the risk that any of these new innovations can cause harm. There are more practical ways to wipe out life.
Yeah. Do you have a final word, Babak?
On that cheery note,
Okay.
so I think this is a field that all three of us are quite excited about. We do recognize that there are potential landmines on the way, and as you mentioned, we have to be very careful about the usage. But I think overall, we're better off knowing these things rather than intentionally shutting down lines of inquiry when we know others will not shut it down and they will move forward. So we don't want to be behind. We want to be fully aware. We want to be actually ahead of the pack, and we want to set the ethical rules and tone of how these advanced technologies are used.